Hello everyone, today we're going to talk about tRNA. tRNA is called transfer RNA. It brings amino acid to ribosomes so that a peptide chain can be formed. Let's recap a little bit. Um, this is a cell and it has a DNA in it. From DNA, transcription occurs, mRNA is produced, and then translation occurs in the ribosome. When ribosome takes RNA into its structure and tRNA, they bring amino acids to the ribosome and they'll bring our proteins, which is right here. So this whole concept is called transcription and translation. And let's discuss structure of a tRNA. So this is a structure of a tRNA. It has 3' and 5' and it's a RNA, right? So it when it get activated with a particular amino acid, so the amino acid is attached right here at one end, and it has an anticodon on the other end right here. These are the anticodons right here. 1, 2, 3. So as you know, one codon specified to one amino acid. So in our case, this codon specified leucine right here. Okay, so that TR and RNA has this anticodon, so it can only attract to its opposite side on the mRNA. Okay, so when it sees there's a attachment that it can do right here on the mRNA, it start to activate the attachment of this particular amino acid to the amino acid chain. So let's talk about how this whole process occurs. Now I have made 3 different steps to show you how ribosome moves from one direction, from 5' direction to the 3' direction. Okay, because it has to read these codons. I have given the numbers to these codons. So first one here, second, third, and fourth. So it's going to move to from 5 to 3 direction. So ribosome has 3 sites. So we have to note it down: A site, P site, and E sites. So I have designed these sites right here: A site, P site, and E sites. So at given time, 2 sites are occupied. So this one is occupied, this one is occupied, this one is empty right now. So at this A site, I have leucine, leucine tRNA already attached here on this side. Okay, and the G is right here, which is glycine. It already attached its amino acid to this peptide chain. Okay. Now ribosome has to move to the next level of codons to read. So this tRNA is ready to move in. So it's gonna move in to this empty space right here. Since this ribosome has moved to this direction, okay, it had occupied the next codon at 4th position, which is right here. Now it's occupying that. A site is occupying this site, and the new tRNA is going to join and make sure that it has a complementary codon on A site, and L is moved to the next location, and G, which was here at the P site, it moved to the E site. You see that's how it moves forward. And once this new tRNA, which is cytosine, is attached to A site, the leucine is already attached here, and look how G is moved forward to his from his previous position. Before it was right here. Now G has attached to another amino acid. Now G tRNA, which was carrying this amino acid at this E position, now it exit out from this E site. So now this tRNA is free to go. So and this whole process keeps repeating. So that's it from me for as far as tRNA is concerned. So recap it: DNA to mRNA, mRNA to ribosome to a protein, and this is the structure of a tRNA. It has an anticodon which attached to a codon on the mRNA, and this is a ribosome. And as ribosome moves forward, this peptide chain keep building up. Okay, guys. If you like this channel, go ahead, subscribe the channel, and give it a thumbs up. See you next time.